Hi, all. Welcome back to Microbiology Lectures, and now we're going to talk about bacteria growth curves in greater detail. A couple lectures ago, I kind of introduced what a bacteria growth curve looks like, but we're going to talk about what a colony, typically a colony in a laboratory situation that's grown in a lab, is going through during these different phases of growth. Okay, so we're going to go through each kind of phase and talk about what's going on, what might be made, that kind of thing. So, closed system, talking about maybe in a test tube or on a petri dish, the waste not removed, new nutrients are not being added. And this is what we'd kind of expect in a situation like that. So say you inoculate a plate or a broth in a falcon tube or a test tube. You would expect the initial individuals to grow and there to be a lag phase. Okay? Again, for most species, it's a couple hour lag phase. And then there's enough nutrients, enough growth. They've replicated their DNA. They started binary fission, and they start splitting, and you get this log phase, this exponential phase, and that goes on for a while, but it will always reach a carrying capacity. They always reach a K, and the carrying capacity will just be determined by, in this case, if it's a closed system, how much nutrients, what kind of nutrients are there, how many individuals did you start with. All of that determines how fast that colony will reach a stationary phase. Okay, so really this is just kind of how a laboratory situation works, but it's nice to know because we can start to look at different things that happen to the colony or happen to the individuals in each one of these phases. Okay, so again, lag phase, the number of cells are not really increasing. They might be increasing a little bit, but it's not noticeable. How many new individuals are coming into the population. Again, this is like a preparatory phase. This is a phase where they might be copying DNA, they might be building new ribosomes, might be copying their plasmids, increasing enzymes, increasing cytoplasm, that kind of stuff. And really, like, how long the delay is will be determined by what kind of conditions the organism's in. Then you have that exponential phase or that log phase, and this is a constant rate of growth. And most of the time, if you take and divide the stationary phase, so the top end, where we might call it the carrying capacity, this line up here, if you divide that in half, then halfway up the exponential phase, that is the part of, or that's the spot at which the time and number of cells at which the population has its highest growth rate. So it's fairly constant growth rate, but there is a spot where you would expect the highest growth rate, and that's half of the stationary phase, or half the carrying capacity. In ecology, when we're dealing with populations that we might want to sample, this can happen in micro also. We want to sample at that phase. It's called the maximum sustainable yield. So the MSY is halfway. So if we were to take that population and we sample it here, then the growth rate is highest, and you would expect them to replace themselves. And so We'll come back to this. It's not important for closed systems because closed systems have a limited amount of nutrients. There's no waste removal, and you can't expect them to live forever in a closed system. Okay, but that exponential growth, fast growth, constant growth, and this is the part where your generation time is measured. So this is part of that equation where how many generations do you have, how many new cells are coming in at a given amount of time. That's really this phase. Okay. The other note that I want you guys to know is this is the most sensitive phase for majority of our antibiotics. So remember, a lot of our antibiotics are blocking different things, or maybe they break bonds, or they affect something in the organism. Like, for example, the penicillins will go in and they'll break the linkage between peptidoglycan layer. Okay? And so as the cells are dividing and stretching, that's a really good time for an antibiotic to come in and start breaking peptidoglycan layers, okay? Because these cells are trying to stretch, they're trying to increase their size, and if they got all these little holes in them, they're very sensitive to that antibiotic, and they don't have a rigid capsule around them often, and they don't have a defense against the antibiotic, which we'll see. Normally that defense, antibiotic resistance, comes later in like a stationary phase when their plasmids start to be more active, and they'll start forming secondary metabolics and things like that. But so very sensitive. Another bacteria or antibiotic like the quinones, they are preventing DNA replication. They block DNA gyrase and topoisomerase. I think topoisomerase 4. I think. Don't quote me on that. But they block that. So both those enzymes a lot of those enzymes are needed for chromosome replication, and so that group of antibiotics will go in and they block the enzymes from working. So you basically can't copy the chromosome, and so great time to use antibiotics is this exponential phase. Okay. Primary metabolics, secondary metabolics, they start to ramp up at this time, like right in the exponential or the log phase. The primaries are important for primary growth. Okay, so the primary metabolites are like proteins, amino acid chains, nucleic acids, lipids, carbohydrates. These molecules that are typically, we know the pathways very well, we know how they're generated. Those are the primary metabolites, and that just helps with growth and allows the organisms to grow and reproduce. The secondary metabolites are coming at the tail end of the log phase, or the exponential phase, or like right before it gets into the stationary phase, and we'll kind of talk about that next. So that stationary phase is typically indicated by low levels of nutrients, so they've already used up most of the nutrients on the dish or in the tube, and they go into the stationary phase. So the nutrient levels are too low for them to increase the population, so they kind of stabilize. You might have a little bit of increase and a little decrease and back and forth, but it's pretty much a stationary phase. But what does increase is these secondary metabolites, and those secondary metabolites, often this is where we see increases in like toxicity, increases in resistance, increases in things like thermal tolerance. And a lot of the secondary metabolites, unfortunately, we don't know that much about the pathways at which they're made. Some of them we know really well, but a lot of the other ones we don't know, like the complete pathway. But we do know that often these secondary metabolites are the ones that allow for these organisms to withstand changes in their environment. So withstand low nutrient levels, increases in temperature, increases in pH, increases in salinity, whatever it might be that the environment's changing. That stationary phase and those secondary metabolites seem to be very important for that phase. Okay? Then you get this kill-off phase, or this death phase, this culling phase, where lots of cells start dying, and they start dying at a constant rate. And because of that death, you start to see a huge die-off, but the nutrients are no longer, the remaining nutrients, the little bit that's left, are no longer being consumed. That's somewhat important because once they come out of the death phase, they go into like a prolonged decline, but it's not drastic for a lot of species. It'll be this like really slow decline where they're just consuming every single bit of the nutrients that are left. Their growth rate is so small. Most of the organisms aren't getting very big, and they just try to maintain their population for as long as possible. So those secondary metabolites are coming into real play here in these phases after they've been generated here. And you can see that normally after the stationary phase, the secondary metabolite generation drops off very quickly, but most organisms have them in the system, okay? Because there's nothing really to build secondary metabolites from. And the primary metabolites, they drop off a lot also, because there's really no source of energy. There's no source of the carbon, the nitrogen, the phosphorus, oxygen. None of that's really available often after you get out of that stationary phase, because now there's just not enough nutrients in that closed system. Okay. So colony growth really depends a lot on what kind of media or what kind of nutrients you give the bacteria, and so how long they might last in a colony, especially if it's a closed system, greatly depends on that. And there are differences between solid media, like an agar plate, and a liquid broth media, but the differences really come in how the organisms are distributed in that kind of media. So if the organism is like at the very top of the liquid broth and can exchange oxygen easier than the ones at the bottom, then those organisms might be growing at a much faster rate than the organisms at the bottom. It depends on their oxygen needs, but knowing that there's a difference means that we can sample the population at different places and get a different growth rate. Okay, so also like on solid media colonies, like in a slant tube or even on a petri dish, the edge of the colony often is doing better than the center of the colony. So the edge of the colony, there's often less competition for oxygen and nutrients on the edges than there is in the center, and so they typically grow better. And you get the die-off or the stationary phase happening in the center, and you can still have exponential growth or the log phase on the edges, and so that's kind of an interesting thing. Same thing with like the liquid broth. You can get the same kind of situation again, like center colonies or colonies in the middle of the media will often deplete nutrients faster. They accumulate more toxins, and like they're driven to a stationary or a death phase faster than the edge. Yeah, so I already kind of talked about this. Again, edges will often show exponential growth while the centers are in a death phase. Okay? So one way to prevent this, and a lot of laboratories do this because they're interested in maybe a bioproduct that comes off of these microbes. So maybe they're interested in an antibiotic that comes off these microbes, maybe an antifungal, whatever it might be. They give something back, or we're interested in something from them. So we'll grow them, or maybe we want to study them for a lot longer under more natural conditions. We'll grow them in open systems. So nutrients are added and waste is removed. And there's a couple ways that we can do this. Chemostat and turbostats are open systems. And like, for the most part, this is just for your knowledge on kind of how these systems work. But often you'll have some kind of flow regulator that's telling you, what the material's like, how many dead cells there are, and whatnot. And when you start building up a bunch of dead material, it will often purge the system in some way. And so you might get a spent culture bottle where all the dead material is coming off, and the live material is still in, like a sampling or a living culture. Okay, and it's the same deal with like the chemostat. It's very similar, except for normally you have this spent outlet where you can just open a valve and remove the material. So some of these are very elaborate systems because we're after cultures that are making some of those, in some cases, some of those secondary metabolites. We're really interested in those secondary metabolites because that's where the antibiotic might be made. And so we'll drive these cultures to go into the stationary phase, but we don't want them to go into the death phase. And that stationary phase, that's where we want them to sit, and then we can utilize them to build antibiotics, and we can test those antibiotics. And then once we get around to it, we can create the same antibiotic in a laboratory without the living organism doing it for us. Okay, so there's lots of things. Again, the nutrient content, the speed of the addition, all that's controlled, so it can give you a constant growth rate, or you can get a system where you're interested in the organisms always being in the exponential phase or always being in the stationary phase. And there's different reasons to do this. So in some micro labs, they're interested in keeping organisms out of the stationary, out of the lag phase, and only exponential phase, so they can study how the organism goes about replication. And this is how we learn a lot about how different organisms replicate, how their chromosomes are replicated, what enzymes are needed to replicate, how the capsule of different bacteria might be formed, and things like that. So you might maintain them in a log phase to grow them and harvest them. So companies that might be sending samples or sending bacteria, and like they might cultivate the bacteria and then send it to other companies, so those other companies or institutions like a college can utilize that E. coli or that staph or that whatever it might be in their labs. They'll maintain cells at the log phase because they can really pump out a lot of cells, and then they'll grab those cells and they'll sometimes freeze dry them or put them on a slant, and you got the best cells possible. You don't have cells in your lag phase. You don't have cells in the stagnant or death phase, but you have your cells that are in that log phase and growing the best they can. Okay. So we're going to stop there. Next time we're going to talk about some things that limit the population's growth, not just nutrients, but other things that are both important for laboratory cultures, but also natural cultures.